All right, hello everybody. Welcome to the final Closer Look Journal Club of the Spring 2022 semester. I'm joined here today by my colleagues Manuel Rausch, Matt Bursey, Johannes Weikenmeyer, and Adrian Buganza-Topole. And we're really excited today to host Colleen Witzenburg. Colleen completed a bachelor's degree in mechanical engineering at Iowa State University and then completed her PhD in mechanical engineering at the University of Minnesota. After that, she moved to the University of Virginia for a postdoc, and then started as an assistant professor in biomedical engineering at the University of Wisconsin-Madison in, was it 2019, Colleen, or? Oh, you're on mute.
Yes, January of 2019.
January 2019, where she runs the cardiovascular biomechanics lab. And her lab, Colleen, does really interesting work towards understanding soft tissue. She pulls from a bunch of different experimental and computational techniques. And as I'm sure we'll see in this talk, she does a really nice job of integrating multiple different experimental and modeling approaches to better understand the cardiovascular system. So I'm really excited for this talk. And Colleen, please go ahead.
Yeah, thank you so much for that really kind introduction, and also for the opportunity to share a little bit about our work. Okay, let's see. I'm really excited to be able to talk about what we're working on. So today I'm going to focus on hemodynamic reflex compensation following myocardial infarction, and specifically what are sort of the implications for ventricular remodeling and hypertrophy and heart failure. The paper that I'm presenting is sort of in this sort of series of papers that we've done on this topic, thinking about the hypertrophy of the ventricle following myocardial infarction, and how do we sort of measure that and model it, and then also what's going on in terms of hemodynamic compensation following a heart attack. I apologize, there's going to be quite a bit of sort of like introduction into this, and so hopefully that'll provide some context for the study that I'm going to go into more detail about today. So I'm just going to start off with sort of the broad impacts of the work. So myocardial infarction and heart failure are big problems in the United States and across the world. There are about 800,000 new heart attacks every year in the United States. Our clinical partners have gotten really good, actually, at treating heart attacks relatively acutely. So 85% of patients who have a heart attack now survive, which is really great. But just because they survive this sort of acute insult doesn't mean they're sort of out of the woods. They now are at risk of heart failure as a heart attack survivor. And heart failure is a pretty common problem in this group. So it occurs in about a quarter of these patients, and it has a really high mortality rate. So you can see here the 1-year mortality rate is really high, and that is not to say that if you make it sort of through the first year, you're fine. The 2- and 5-year mortality rates are also quite high, and so we really wanted to start thinking about what are sort of the long-term implications of this heart attack in terms of cardiac function, and we're thinking about it really from a mechanical perspective. So a heart attack causes this primary reduction in function in the infarcted region. So if you're not familiar, the downstream heart muscle from the blocked artery dies, right, in response to this blockage, and it is not replaced with healthy active muscle, but with non-contractile scar tissue. So if we look here, we can see this is an MRI of a mouse prior to the induction of infarction through ligation of the coronary artery, and then 2 days following that myocardial infarction. And so you can see with late gadolinium enhancement that we can sort of visualize where that injury occurred in the ventricle. So this is that primary reduction in function that we talked about. There's also this secondary reduction in function that occurs sort of over the longer term. And this time-dependent secondary reduction in function is driven by hypertrophy of the non-infarcted tissues. So we can see, even 14 days post-infarction that the scar region is starting to form, but also that the cavity volume of the ventricle is increased. And if you did some segmentation here, you would see that the overall amount of muscle here has also increased. And so when I first started looking into this problem and talking about this problem, I thought, well, that sounds great, right? You lost a certain amount of muscle due to this heart attack. If you have this time-dependent hypertrophy where the remaining muscle gets bigger, shouldn't that that be good? Shouldn't that help you sort of solve this problem that you've lost contractile muscle, and your body doesn't sort of reduce its demands as your heart is injured, right? So this is the thing that we didn't talk about, which is that just because you lose 20 or 30% of muscle doesn't mean that you can suddenly stop producing the pressure and cardiac output the body demands. And really the concern is something called this eccentric hypertrophy. So as these muscle cells are growing, if they do so along the circumference more than they do so along the thickness, what happens is that the cavity volume starts to increase, right, with that increase in muscle mass. And so that ratio of sort of cavity volume to wall volume or muscle starts to get out of whack. And this becomes sort of a cyclic problem, right? You add a little bit more muscle along the circumference, that increases the cavity volume, which causes you to increase more muscle. And you end up with this eccentric pattern of growth, which eventually devolves into heart failure because the ventricle can no longer produce the cardiac output the body needs. So this is a failure in the systemic the systolic function of the ventricle. And so when we think about heart failure, this sort of precipitates that final clinical issue where you've got this reduction in cardiac function. And so it sure would be nice to be able to figure out and predict which patients are going to exhibit this sort of pattern of eccentric hypertrophy and heart failure. So this group looked at sort of what happens in terms of eccentric hypertrophy in patients who've had a heart attack, right? And so what they did, I think, is really nice. So they did cardiac MRIs in patients who had had a heart attack within a week of their initial event, and then 3 months later, and they just quantified the change in this cavity volume, or this LV end diastolic volume over that period of time, and plotted it as a function of a lot of different predictors. And sort of the only predictor that they found that was significantly correlated was this infarct size. So how big was your heart attack? And so that was significantly correlated with this dilatation and reduction in cardiac function. But I think when I looked at this plot, what I really saw was a lot of variability, right, in patients with infarct size, right? So this is the best predictor, and especially if we focus sort of on these mid-size infarcts, so anywhere from, maybe 7 to about 40% of the heart muscle loss. It's really unclear, right, based on infarct size alone, whether these patients are going to exhibit dilatation or not. And so, we're basically seeing this rather large variability in post-infarction ventricular dilatation. And so as modelers, one of the questions was, well, okay, can we predict ventricular dilatation or this hypertrophic pattern using our computational model to start to try to understand why this is happening? And maybe we can then identify patients that are more likely to exhibit this pattern and have heart failure. So we built a little computational model. This is a relatively simple model. It's the circulation is a lumped capacitance representation here, and our ventricle is a spherical representation of the ventricle. So very simple in terms of the components here. And when we use this model to represent or to model a couple of different pathologies, so we looked at volume overload, and we simulated that with by putting this resistor here to simulate mitral regurgitation, which is what we had data on. And typically the pattern of growth following volume overload is this eccentric growth pattern, right, where the ventricle increases muscle mass, but primarily along its circumference. We also modeled aortic constriction or pressure overload using our model, and typically this causes a growth pattern where there's thickening or increase in the muscle mass, but no change in the cavity volume or small changes in cavity volume. And we can think of this as sort of diastolic dysfunction, whereas volume overload tends to cause systolic dysfunction in the ventricle. And then finally, we used our model to actually simulate myocardial infarction by sort of separating our ventricle into an active compartment and a passive compartment according to infarct size. And so then within our model, we also embedded a growth law, so we could actually simulate changes in ventricular muscle mass over time in response to hemodynamic changes. And we did this based off of a strain-based growth law. So our growth law takes changes in circumferential strain, and those cause dilatation or shrinkage of the ventricle. So you can see here how we're computing circumferential strain. We take the peak value during the cardiac cycle as our stimulus for growth. And so if this increases, you can see that the diameter of the ventricle increases, but its thickness stays the same. So overall, there's an increase in muscle mass, but it is an asymmetric increase. Changes in radial strain induce LV thickening or thinning in the model. You can see here again, we're taking peak radial strain throughout the cardiac cycle, subtracting off some set point. And these changes, so here, if it increases, we would see a thickening of the ventricle and no change in its diameter. And so we used a couple of different literature-based studies in dogs to fit these parameters and get our model going. And then once we had our model fitted, then we validated it against 3 other data sets. And so here are the results from those 3 other data sets. And so you can see, generally speaking, we are doing a really good job of predicting these changes in dimensions over long periods of time. And here I'm saying, sort of days, weeks, months in terms of these predictions. So the model predictions are shown in green and the data here is shown in gray. And we were really excited about this result. But the thing that we sort of started to think about and notice was what was really important for the success of our model. And so what we found is that these acute changes in strain, right, what's happening when you make these large hemodynamic changes, were really critical in predicting growth in terms of, especially in terms of trends, right? So in volume overload, we pretty much always saw that circumferential strain increasing, right? So this led to an increase in cavity volume. In pressure overload, we pretty much always saw that radial strain increasing. And so this led to an increase in thickness as we looked at a variety of experimental data sets. The challenge was myocardial infarction. So part of the challenge was we're just looking at the surviving myocardium, so it's more challenging to sort of estimate what strain is. And then also we were seeing just a lot more differences in the hemodynamics as we looked through different dog studies. And so this really led us to sort of ask this question of, okay, what's going on acutely hemodynamically in response to infarction? What we discovered and what we were working on was this idea that there's an acute physiologic response to infarction that involves hemodynamic compensation. So in the model, if you have your baseline model working well, and then you just simulate infarction and you don't change any other parameters, what you see is this nice classic rightward shift of the pressure volume loop, which is something that you see in both animals and humans, right? But what is not what you see in experiments is this rather large drop in LV pressure, right? That doesn't happen in humans and animals. And that's because in organisms, right, they compensate post-infarction to restore or maintain mean arterial pressure, right? The body says, hey, I need a particular pressure. And so there are these different mechanisms that kick in to keep that pressure at a particular level. And so this includes heart rate, contractility of the surviving myocardium, arterial vasoconstriction, as well as venoconstriction. So these mechanisms work in concert, right, to restore that mean arterial pressure. And so we really wanted to sort of look at, well, what does this look like in myocardial infarction, right? Are these related to infarct size in some way? Do they kick on sort of one at a time? Is there some predictable balance in how this is happening? And what are the implications for strain and then ventricular hypertrophy? And so sort of a sneak preview of this is one of our questions was, is there a lot of variability in this hemodynamic compensation, right? So that was sort of where we were starting here. And so we used our model to actually fit some of these parameters to an old literature studies in dogs. And so some mechanisms can be directly measured, right? We can measure heart rate, arterial constriction or systemic vascular resistance can be estimated from measured values, and other mechanisms can't be measured. So we can't measure venoconstriction particularly well. And then contractility of the surviving myocardium is not possible, right? You can measure contractility of an intact ventricle, but as soon as you've got the infarct, it becomes much more challenging to actually measure contractility. And so we actually use the model to fit these two parameters based on measurements that could be made, namely LV end diastolic pressure, pressure generation, and then mean arterial pressure. So here are those modeling results for that historic dog data. So We really like this study because they reported individual animal-specific data, which was really nice, right? So we have infarct sizes and hemodynamics for each individual dog. And you can see that in comparison to sort of the sham group, these are the heart rates and arterial constriction values. And there doesn't really seem to be much patterning here. We were really surprised that some mechanisms actually decreased post-infarction. And then when we look at contractility and venoconstriction, we sort of see a similar response, right? That there's a lot of variability here, and there are no sort of obvious trends in this data and in the modeling results. So then we asked, does this variability in compensation actually lead to variability in predicted ventricular hypertrophy from our already validated model? And so we input this into the model and predicted the change in end diastolic volume and thickness of the surviving myocardium over time. And what we saw was somewhat exciting in comparison to the human data, right? Where at low infarct sizes, we didn't see much dilatation or really much change in thickness. But as we moved into sort of these mid-level infarct sizes, we saw a lot of variability, particularly in dilatation of the ventricle. And so, we thought, okay, this is a plausible cause for all of this variability that we're seeing in post-infarction hypertrophy. So some key points here: we saw a strikingly wide range of compensatory reflex profiles. We think that this could potentially translate into post-infarction variability and dilatation. And so that's sort of the hypothesis that we were starting with. Now, we had some concerns. At this point, all we had done was modeling and looking at some old historic data. And the main concern that we had was in sort of this variability in hemodynamic compensation. So people were asking us, well, maybe this is just variability among animals, right? Maybe animals just have a lot of natural variability in these metrics, regardless of their post-infarction status, right? So pre-infarction, there's a lot of variability. And so your comparison to the sham group maybe is sort of unfair. And that's really part of what motivated the study that I'm going to talk about in more detail from here on forward, which is we wanted to do this prospective study in individual animals to really robustly ask this question, right? Is the variability actually in hemodynamic compensation? So we're going to have animals service their own controls. We're going to really fit the data for each individual animal really rigorously. And then secondly, the question was, okay, well, if animals do need to service their own controls for you to make these distinctions, how are you going to go ahead and detect this balance, right? Because in most cases, you're not going to have control measurements in humans, and maybe not even in experimental animals. So our question was, can we use pharmacologics to see, okay, if we can detect how a particular animal did respond to infarction? And so those are the two sort of hypotheses that were driving this study that I'm going to talk about. Okay, so we performed our study in adult male Sprague-Dawley rats. We looked at left ventricular segment lengths in order to compute left ventricular volumes, as well as measuring ventricular pressures and heart rate throughout the experiment. And we did this at multiple time points. So for each animal, we took these measurements at a controlled time point prior to ligation. Following ligation of a coronary artery, so we had sort of a myocardial infarct measurement time point. Then we administered either dobutamine or nitroglycerin, took a second measurement. We waited for the drug to clear and took a measurement of MI again. And then we administered the other drug, right, nitroglycerin or dobutamine. So why select nitroglycerin and dobutamine? So we specifically wanted to target compensatory mechanisms that we knew we couldn't measure, right? So that contractility of the surviving myocardium and our venoconstriction. So we picked drugs that dobutamine is known to affect contractility, and nitroglycerin is a venoselective vasodilator. So it has a stronger effect on the veins than the arteries. And so that was one of the driving factors behind these two pharmacologic choices. Secondly, we were also looking for high clearance rates. We wanted to administer both drugs to every rat. And then we were thinking about, if this is going to be a biomarker, or we're going to try to evaluate this, we want to be able to do it quickly, right? And see the response really quickly and not have a long-term effect on the hemodynamic balance. just briefly, we allometrically scaled our dog model to rats because we moved from dogs to rats here. And then we directly determined our LV active and passive behavior in the model from IVC occlusion data. And I forgot to say this earlier, but at both the control and MI states, we did IVC occlusion. So we occluded the inferior vena cava to make sure that we had a really nice set of data to fit these active and passive properties. So we could get those in an animal specific way. For heart rate and systemic vascular resistance, we basically did the same thing that we did with literature data, right? We just input the measured heart rate. We computed systemic vascular resistance from other measurements, mean arterial pressure, stroke volume, and heart rate. And then we fit our venoconstriction and contractility metrics, stress blood volume, and elastance in the model by just minimizing error in those end diastolic and end systolic pressure volume points. Just as sort of a note, we did also fit the ratio of our 3-element Win-Kessel resistances, so to minimize error in pressure during ejection. So if just to say, if you wanted to talk more about model fitting, I'm happy to go in the details, but I figured that's probably enough for now. Okay, so here is the model fit for a representative animal's data. So the data here is shown in the circles. Model is the lines. And so I hope you can see that our model is fitting the data really, really nicely, both before and after infarction. This animal received nitroglycerin next in their workflow. And so this is the data and model for the nitroglycerin step, as well as that second MI measurement, just to validate that, yes, the drug had actually cleared. And then finally, here's the response of this animal to dobutamine, as well as the modeling result for dobutamine. So really now we wanted to get into the compensation, right? How did these different animals compensate? So here for that representative animal shows their balance of compensation. So this animal compensated primarily with systemic vascular resistance and contractility of the surviving myocardium, and actually exhibit a decrease in venoconstriction. And if we look at all of the animals from our study, each animal is a different color. We can see all of the sort of reflex balances of these different animals. And I hope that you can see here that there's quite a bit of variability in these responses, right? So all metrics had some animal have a reduction in that particular metric. We have a pretty wide swath, especially when we look at contractility and venoconstriction. On average, there was an increase in systemic vascular resistance in our rats, but there weren't any average changes in sort of any other metric here. So now that we had this animal-specific information, we could actually analyze these relationships to see if they were correlated with one another, right? Or if they were correlated with infarct size. So if infarct size was driving an animal to a particular balance. And the only correlation that we saw was between venoconstriction and contractility. We didn't see any correlation with these metrics with infarct size, and we didn't see any other correlations between them. And we were a little bit surprised by this, particularly for venoconstriction and arterial resistance. We thought that those would be likely correlated. And so we were, a little bit surprised that we didn't see a correlation, but we didn't. And so here that we do see a negative correlation between venoconstriction and contractility. So animals that exhibited a decrease in one tended to exhibit an increase in the other. So that was the result there. Then we move forward to thinking about these pharmacologics. So this is just showing the change in these different responses relative to where they were immediately post-infarction. So the question, right, of course, is, did nitroglycerin perturb this balance? And so we expect nitroglycerin to reduce stress blood volume, right, because the veins are going to dilate. What we didn't necessarily expect was that this was going to be accompanied by a large decrease in mean arterial pressure, right? So contractility and heart rate did not dial up in response to this nitroglycerin administration to maintain mean arterial pressure. And so that was sort of one thing that we had not sort of anticipated. And then the other thing we wanted to know is will this be a predictor, right, for, or can we use this to sort of go back and see whether or not these animals relied on a particular mechanism, and in this case, venoconstriction, right? So animals that venoconstricted a lot, we thought maybe would be more sensitive to dobutamine or excuse me, nitroglycerin, or less sensitive to nitroglycerin. And what we found was that there was really no relationship between their initial response in venoconstriction and their response with nitroglycerin, right? So there's no correlation between those two, at least at this particular dose. Next, we looked at dobutamine, and we were buoyed here, right? We know dobutamine should increase contractility. We see that increase in contractility in every single one of our animals with this administration. We also saw small but significant increases in heart rate and mean arterial pressure with dobutamine administration. In this case, we thought, we hypothesized, right, that animals that had already compensated strongly with contractility would have less of a response to dobutamine because their contractility was already sort of high. But what we found again was that there was no correlation between this change in contractility with dobutamine and that initial response to dobutamine. So, if we're going to try to use these pharmacologics to sort of tell us how this animal initially compensated, as far as we can tell, that there doesn't seem to be that doesn't seem to be a great strategy for that. So we started this conversation talking about post-infarction heart failure, right? And so the next thing we wanted to ask was, okay, well, what happens to the strains in these animals? We know that strain is linked to hypertrophy in the model. And so how do these strains change? So this is showing the change in peak circumferential stretch with infarction, as well as the peak radial stretch with infarction. In general, we saw increases in peak circumferential stretch, which, remember, is correlated with that eccentric hypertrophy sort of pattern, and no overall difference in peak radial stretch. If we looked at the levels of these values that were necessary to produce enlargement and thickening or thinning in the dogs, right, because we're now in stretch, and so this is a non-dimensional parameter, what we see is that enlargement would be predicted for 6 of the 13 rats. So we would have predicted an increase in cavity volume in about half of our animals, whereas thickening and thinning were really only predicted for 1 of each animal. And this was sort of consistent with what we had already done in our modeling in dogs. And so in some ways, this was comforting in terms of like, okay, the stretch seems to be doing what we think would make sense given all of the results that we've had so far. So our next question was, did we see changes in stretch or correlations in stretch with other metrics? And so we looked at infarct size in particular, right? Was infarct size correlated with this change in peak circumferential stretch? We found it was not. So we didn't see any correlation between what we think is our hypertrophic stimuli for eccentric hypertrophy and infarct size. We did see a sort of weak but significant correlation with venoconstriction. So as animals used venoconstriction at higher amounts post-infarction, they were more likely to see increases in this circumferential stretch, and potentially more likely then to exhibit eccentric hypertrophy. Okay, so sort of some concluding thoughts on the work that I've presented here. So that one of the big questions that we had was, is this variability in hemodynamic compensation that we're getting from sort of literature data and some computational modeling, do we actually see that in real animals as we start to look at this prospective study in rats? And I think that we have really good evidence now that that's actually happening, right? So when we really robustly fit our data with the model, we see that these individual animal responses are highly variable post-infarction. We also had some other observations. First, that venoconstriction and contractility were negatively correlated in our study. When we went back to the older dog data and started to ask questions about these 2 metrics, we did see that they generally decreased or increased together, right? So that this relationship seemed to be reasonable given the data that we had looked at before. And then as we were looking through sort of other individual animal specific data, it seems like that's consistent consistent. And so we don't know necessarily what the implications for this are, if this means that maybe animals who have some damage to their surviving muscle have to then rely on venoconstriction, or if venoconstriction is telling us something about that. But we think this is an interesting result. And then finally, we didn't find infarct size to be correlated with compensation or with stretch, which we weren't really expecting, but is going to make moving forward, I think, pretty challenging as we think about how do we model this and how do we estimate individuals' hemodynamic responses, because we know we can measure infarct size both in an experimental and a clinical setting, and so I think we were sort of hoping or looking at that and thinking maybe there would be some relationships there, but we really didn't see any in our experimental study. So then the second part of the study was really asking, can we detect these balances post-MI using pharmacologics? And largely, we didn't see that was happening. There was no correlation between drug treatment and compensation that we found, particularly as we looked at compensatory mechanisms that we knew that the drugs were targeting. And we used relatively low doses, right? So our dose of dobutamine was a fraction of what you would give in sort of an exercise type test. And then MAP was really, really sensitive to these pharmacologics. So one of our thoughts was, well, maybe if we could give low enough doses that we didn't see large changes in mean arterial pressure, we might be able to start to get at these correlations. But I think that overall, this is going to be a pretty challenging approach, just because the dose response in animals is going to be a hard thing to sort of overcome. And so maybe as we look at sort of predictors or trying to estimate compensation, other approaches may be more beneficial, right? So going through and saying, can we look at acute volumes and use those to try to estimate what stretches, rather than spending as much time thinking about pharmacologics in this space. So a little bit of and I also think that in our study, right, we were able to show that variability in this predicted post-infarction ventricular dilatation was precipitated by hemodynamic compensation. So that was really driving this variability in our predicted ventricular dimensions. And just a note on that, we've been working on that a little bit in more detail here. So we looked at a study where they administered nitroglycerin chronically to dogs that had infarctions. And so the control dogs here are shown in gray. This is their data of end-diastolic volume change. Those that were given nitroglycerin chronically are shown in this purple color, and this is their change in thickness. Unfortunately, they didn't record acute hemodynamic changes, and so we had to pull those from a different study. So that's probably why our modeling results are not matching these quite as well as they could be. But we were able to see in the model, right, if we tried to replicate these acute changes with nitroglycerin and then hold them moving forward, that we could start to replicate these differences in ventricular changes. And so I think this highlights, first off, that there are differences in ventricular hypertrophy in animals that are pretty large, and that at least we can see acute differences in hemodynamics can start to replicate those within our model. And so it's plausible that we can sort of get at this question in a modeling perspective on how hemodynamics are playing into long-term hypertrophy of the ventricle. So that leads me to sort of the last point on this, which is, I've been talking about predicted post-infarction ventricular dilatation. And this is, I think, one of the areas we really want to target moving forward is getting rid of that predicted and running experiments where we actually measure post-ventricular dilatation over time. This can be really challenging to try to quantify both hemodynamics and ventricular dimensions, both acutely and then over time. It's an expensive experiment. It's challenging to measure pressure multiple time periods. So we're working on it. We're trying to really bolster up our modeling results to say, yes, we think that this is happening before we can sort of justify getting into a rather long and expensive experiment. But that's sort of where we're going with this. So I want to acknowledge the wonderful people who helped with this work. So these are some images of my group here at University of Wisconsin-Madison. And then also a lot of this work was done sort of at the end of my postdoc and the beginning of my faculty career with some collaborators. So Laura Caggiano, who's now at UC Irvine, did all of the animal surgeries that I talked about today in rats. And then Jeffrey Holmes was my postdoc advisor and is now the Dean of the College of Engineering at UAB. And then I also, want to acknowledge our funding sources. As with starting a new lab, it's always challenging to find funding. And so we've been fortunate to be supported by a number of agencies. And so that leads me into my last slide, I promise, which is on other research, right? So I've been talking a lot about infarction and hemodynamic compensation. Our lab also has some other thrusts in research. And I would be remiss if I didn't talk about those. So Ashley Hibbing, who's a graduate student in my lab, has been taking the model in a new direction and using it to model hypertrophy in infants with congenital heart defects. And so she's been starting by asking questions about somatic growth, right? So their ventricular growth is not always bad. It's often good and not pathologic. And so she presented a nice abstract on her work at the AHA scientific sessions this past fall. And has currently got a manuscript in review on that work. So hopefully that'll be out relatively soon. And then Daniel Pierce, who made both the pictures here, is another graduate student in my lab, and he's working on quantifying the material properties of the ventricle, specifically thinking about, okay, we've been talking a lot about what's going on in the healthy muscle, but have largely been ignoring the scar. So quantifying what are the material properties of the scar. And so this is some work that he's going to be presenting at SB3C, looking at quantifying material properties of tissue analogs in a couple of weeks. And then he'll also be presenting some other work that we did in collaboration with a group looking at different treatments for heart valves, where he's done some quantification of the material properties of heart valves. So just wanted to also say, there are other exciting projects that are going on in our lab that are, still sort of in these early phases, but that we're starting to make some progress on. So with that, again, I want to thank you all for your attention and encourage anyone to feel free to ask questions. I put my email and website at the bottom. So if you're someone who, sort of ponders things and then comes up with a question later on, feel free to contact me.
All right. Thanks, Colleen. That was great. Does anyone have any questions?
Sorry if I missed it, and I'm going to just ask a very practical question. Who ended up doing the experiments with the rats? Did you do that in your lab yourself?
Yeah, so I did the experiments at UVA when I was still a postdoc, but the funding for it was all through a little grant that I had written, and so we paid for. So it didn't cover my salary, but it did cover all the rats. And then I was fortunate enough to have access to this amazing technician in Laura, who was willing to do the work for free, which was awesome. She's now a lab manager at UC Irvine for Naomi Chesler, who runs the Cardiovascular Research Center there. And I learned very quickly that it takes a lot of skills to do these experiments.
Yeah,
so
no, that's really fantastic work. Congratulations. And I was curious, whether you did only the analysis or whether you did
yeah. So she did all the surgeries and I was there sort of side by side, like monitoring the pressure catheter and hitting the buttons and making sure that we got reasonable looking data, but was not the one with the scalpel for sure.
But very impressive, Colleen.
Oh.
Looks like we have two questions. Mridang, you're at the top of my list on my screen, so maybe we'll take Mridang first.
Thanks, Matt. Thank you for that great presentation. So I have a question about the animal model that you used. Is there a reason why you excluded sex as a biological variable in your study?
So I'm going to unabashedly say that it was primarily a financial reason. We know that male and female humans and rats have different responses cardiovascular wise and different risks in terms of cardiovascular issues, especially myocardial infarction. So I would have loved to, double the number of animals and been able to look at sort of males and females. But we just, the budget wasn't necessarily there at that point. The other thing that I really, looking back, wish that we would have maybe thought a little bit more about was increasing the age of our rats. And this is sort of a pet peeve of mine as we've been looking through the literature is a lot of folks, including ourselves, were inducing infarction in relatively young rats, right? And you don't see a lot of heart attacks in 20 and 30-year-olds. And so we should probably be using older rats as we start to study these things. But hindsight is always 20/20.
Thank you.
Craig, you want to go ahead and ask your question?
Perfect. Hey, Colleen, great talk.
Hey, thanks for
How are you?
coming.
Yeah, no problem. It's late here, but want to stay up for it. Feel you on the variability in the animal models. That happens for us too, and it's something we deal with and struggle with frequently. So with the longitudinal study, you talked about how that would be difficult, but can you maybe expand on that a little bit and say, what types of measurements would be needed? Could you do it with a non-invasive approach, or would you have to get animals for every single time point and do an open procedure and terminate?
Yeah, that's a great question. So we've been thinking about this a lot. So in terms of volumes, right, that's not a challenge. Non-invasive imaging, whether we do MRIs or echoes, right, we could do that over multiple time points. The real challenge for us is pressures. So we haven't been able to get through the model without a left ventricular end diastolic pressure. And as far as rats go, we don't know of a non-invasive way to get at that pressure, right? If you're in larger animals like dogs and pigs, you can do a catheter and get a wedge pressure. So if you know a way to get left ventricular pressure in rats, there's this crazy group that has implanted it into the left ventricle and left it over periods of time. And so if I can convince that PI to send me their little device, I will do it. But the challenge is as the animals grow, left ventricular pressure is not just a function of material properties, but also a function of that growth, right? So that passive relationship that we see in the pressure volume curve is dependent both on dimensions and on material properties, which are both changing over time. Right. So just having the volume isn't, so far we haven't found a way to make that sufficient. And so that's really I would say the number one stumbling block we're coming up against.
Okay. Yeah, some telemetry device of some kind that would be implanted and then left, and then sure, it'd be expensive and tricky to put in, but okay. Yeah. Cool. Well,
if you know of a person who's doing these telemetry devices who is excited to send them off, and I would love I'd love the contact.
I'll ask around. I'll let you know.
Thank you.
Thanks.
Okay, any other questions?
I have one.
Yes.
Yeah, sure. Great talk, Colleen. Really appreciate this, and this also makes me scratch my head on a lot of these things that I would also expected to have had more of a correlation there. Just now that we're talking about the pressures, for the pressures that you did measure, what were the challenges in doing that? Because I've looked at some data in the past as well, and just the pressure measurements on their own were already very showing a lot of variability, and I was just wondering what your experience with that was.
We did see some variability in pressure measurements between animals for sure. I think the thing that I learned was we had to be really careful about zeroing our pressure catheter and making sure that we turned it on, hours ahead of time so that it could warm up. And we always zeroed it on the, the table next to the animal every single time. We took sort of pressure measurements under fluid before we stuck it into the animal. So really trying to just make sure that the catheter was reading properly before we got into the animal. At least for us, those pressure catheters were really touchy. And so that was really important into sort of saying, okay, yes, it's working. Yes, it's giving us readings that make sense. Beyond that, I mean, I don't know that we had we were sort of expecting variability in lots of different ways. So I can show Let me show. So this, when we were fitting ventricular specific parameters, this is just showing that IVC occlusion data. And so that enabled us to then fit, or I should say, yeah, then prescribe the control values of elastance and that intercept there. And so I think that was the other thing that I would say was really beneficial, was doing those IVC occlusions just to see, okay, are things going the way we think they should be going, and do these data make sense as we started to think about pressure measurements, in particular for the end diastolic pressure volume relationship. You can see that the data is pretty clustered for the different points and drug treatments. And so that having that IVC occlusion was really helpful. We tried to do an aortic occlusion to get the sort of other end of the curve. And it worked in some animals and was less successful in other animals. And so ended up not including that data in our final study.
All right, thanks.
Yeah.
So I had a question. So you talk a lot about the hemodynamic measurements and obviously the complexities associated with the surgical approach and everything, and actually just getting these measurements. But I was curious, did you look at any of the tissue afterwards to see if there was sort of structural changes that you could maybe correlate or relate to, some of these hemodynamic measurements? More interstitial collagen or something that may be related to decreased contractility?
Oh, that's a good question. So I should say this is an acute study, right? So they get the infarct, we do the study and then sacrifice the same day. So I'm not going to expect changes in hypertrophy or collagen content due to that infarction. But if your question is sort of like, are there natural variations in these things that we could have quantified? That's clearly a missed opportunity, right? We probably should have taken that tissue and sliced it and done something with it afterwards. I honestly can't remember. I think we threw it in formalin. So I could maybe go back and say, hey, can we do some sectioning and try to quantify collagen content or sarcomere size or dimension. We did make sure, so when we sacrificed the animals and we pulled out the heart, we did take images, or we filled it with agarose and took images of where the sonomicrometry crystals were, so that when we did our volume estimations, if the crystals ended up in a really odd spot, which Laura was amazing, so they almost never did, but if they did, we could make corrections on that if we wanted to. We didn't end up needing to, because she had really gotten the surgery down, thankfully, but we had that information if we wanted to try to get at that value. At least visually, we didn't see large changes between hearts. We also measured the weight of the left and right ventricles, and we used that as we were estimating infarct size. So we did retrograde perfusion of the ventricle with these fluorescent microspheres in order to quantify infarct size acutely. And so then we did slices of the ventricle in the short axis direction and took images of those to actually get at infarct size. So I would say acutely it can be challenging to do infarct size, because you don't have, unless you're doing late gadolinium enhancement and the animal is still alive, but you don't have like scar tissue to be like, oh, there's the infarct. So you've got to think a little bit differently about how to quantify infarct size.
Yeah, I guess, yeah, that's all great. I mean, I was just kind of thinking about what could be the source of some of this variation in animals that are otherwise should be similar. I wonder if there's something, but I don't know.
Yeah, I don't know. My goal, I think at my point, I'm trying to embrace the variability rather than minimize it. Understanding that like humans are going to come into this space with a crazy amount of variability, and so so our goal is really to sort of ask, like, is this variability telling us something? What does this variability sort of mean in the long run, as we try to figure out like which patients are going to end up with this pathology and which patients are going to be fine, right? That was really my thought, was like, okay, rather than saying variability sucks and how do we get rid of it, like how do we embrace it and ask questions about like, is it telling us something that then will, like, does variability early on magnify and become variability in the long term, is really sort of our thought process.
All right, so there's a question in the chat from Ryan. I will assume that he doesn't want to ask, so I will, I guess, read it to you, although you can read yourself. But how does the model account for differences in coronary vascular structure? And do you expect that would make a difference in growth, either between species or even differences between animals?
Oh, so the model doesn't account for differences in coronary structure basically at all, right? We just sort of input an infarct size. We've talked about this in the model. We largely as a question of free wall or septum has been our discussion, right? So if the infarct is in the free wall versus the septum, that could have implications for RV function, which then could sort of precipitate into LV function. And so we've been talking about changing the geometry of our model to more of a triseg approach, which basically is slightly more complex than a sphere. It's like two spheres glued together as a septum. I really like that approach in terms of modeling. I'll say, for growth modeling, if you try to use a really complex geometry, it really kills you in terms of computational power. And so we've been really focused on trying to keep our geometry as simple as possible. But that is one place where we feel like maybe we need to be more diligent, and the ability to have a septum could be helpful as we think about like whether or not the infarct is in the free wall versus the septum. As far as between species or between animals, again, we're sort of embracing variability here. We purposefully tried to induce a variety of infarct sizes because we were curious, right? Does infarct size change compensation? We also purposely didn't do massive infarcts. So when you look in the data on rats, what you'll notice is that a lot of studies will throw out infarct sizes like less than 60%, right? Because there's so much variability in that group. And so because we were interested in variability, like we sort of avoided the massive infarct sizes. And when we do our modeling, what we find is when the infarct size gets to be really, really, really large, venodilation is like the only mechanism that can restore mean arterial pressure in those cases. And so that might be why you see hypertrophy in that group, or it may be a precipitating factor of hypertrophy in that group, right? That they've sort of either saturated all of their other mechanisms, or like that's just the only thing they have left to keep mean arterial pressure high.
All right, well, hopefully that answers his question.
Yeah.
Anyone else? Any other questions?
I just want to reiterate that I really like the work and it's very impressive.
Thank you.
This stuff is hard, and just doing such a comprehensive analysis, including animal work. And yeah, anyways, I loved it. Thank you for sending it.
Great. Yeah, I also, I would kind of comment, your previous explanation of the simplified geometry, oftentimes we, for those of us who do computational mechanics, often tend to go toward maybe more complex models. And I think highlighting here the utility of simplicity is a good thing. And, when applicable, maybe we can start to think about that.
Yeah, I spend a lot of time thinking about boundary conditions rather than the geometry, right? So if you think about hemodynamic compensation, it basically, in a lot of ways, is just boundary conditions, right? Like what pressure is this thing pumping against? What volume is coming into this ventricle? And the thing that struck me as I was working on this was how there's a lot of variability in boundary conditions, and how do you set them? And they're going to change as soon as you have pathology, because the circulation is going to respond to this fact that you have an injury. And so I think that has sort of where we've gotten stuck in thinking about more complex geometries is as soon as you want to start to ask questions about variety and boundary conditions, you just get killed by the computation times.
All right. Any last comments?
I was gonna say I'm gonna stop recording, but I'll repeat Manuel's comment. That was a really nice talk, Colleen, and it's nice to see what you've been working on, and I'm glad you included a slide on some of the upcoming stuff too.
Yeah, the pandemic has made, I think, sharing our work a little bit more challenging. I don't know if your experience has been the same as mine, but my student, who got into AHA scientific sessions was really excited to present her poster. It had like one person come, and it was such a bummer, right, to have this virtual conference that where it wasn't necessarily well attended, and maybe not, like they hadn't quite figured out how to organize it. And so I'm excited to have in-person conferences again. I also think, for students, sometimes going to a conference is like a mini vacation, right? Like it's really exciting to get to go to a location and, stay in a hotel with all your friends. And so like she's sort of been missing out on those opportunities. And so, yeah.
Yeah, definitely figuring out how to share work, and especially work that's like sort of on the verge of being published is like what are the mechanisms now for that?
Yeah. So I also just wanted to thank you again for inviting me to share our work on this virtual platform. I'm really excited. I've been coming week after week. It always seems really well attended, and people ask really great questions. And so, if I was like, if I'm going to do a virtual format, this was the one I was like, I will do that one, because it was just really nice to have one where there was interactions between people, and it felt more like the seminar and less like sort of talking to an empty room. Which, I don't know how many of you taught virtually, but man, that was like pulling teeth to get students to ask questions.
Wow, those may be some wonderful